but it's morning over here for me, so 9:15, not too late. Thanks for having me. My name's Tyler Ciernak. I am an assistant professor at Georgia Southern University, which is based in Savannah, Georgia, on the coast here. And today I'm going to talk about some of my work on coral reefs and carbon fluxes in reef ecosystems. So, so the idea of what makes a coral reef functional and healthy, is different depending on who you talk to, what field they study. But generally, one concept really comes up consistently throughout them, which is the production of calcium carbonate is critical. And when you think about reefs, this is important for a myriad of ecosystem services, but really creating the habitat that then allows these reef ecosystems to support nearly 30% of the biodiversity of the oceans is the thing that's really making these systems functional. And that's the structure that provides uh, protection against wave action and things like that to coastal communities. So it's really what's creating the important base of coral reefs. And it turns out that coral reefs produce about 50% of the calcium carbonate in shallow neritic coastal oceans, and somewhere around, depending on the estimate, up to 15% of global calcium carbonate production in the ocean. So they're quite important on a global scale. So this is a paper that I wrote a while back in 2018, where I was trying to take the metabolic pulse, so trying to use calcium carbonate chemistry data from coral reefs to understand more about their sort of metabolism and the general state of their metabolism, and really the ratio of calcification to production. So on a reef, you can sort of break metabolism down outside of what's going on in the sediments into these two categories, which is inorganic calcification, which you can see here with calcium and carbonate precipitating into calcium carbonate, or vice versa, where it dissolves. And then you've got net ecosystem production, where you're creating organic matter through photosynthesis or degrading it through respiration. And these two things impact the carbon system quite differently, where organic processes change DIC, but they don't change alkalinity. And then inorganic processes change DIC by 1, and they change alkalinity by 2. And we can use these ratios to really look at what's going on in coral reefs from a biogeochemical perspective. So that's sort of the baseline. This study was aimed at taking data from a bunch of reefs around the globe and trying to put together some sort of meta-analysis where we were trying to understand, can we find anything about reef metabolism and biogeochemical function just by looking at how these two processes, or how these two components of the carbon system, alkalinity and DIC, are related to each other. So you can see across the reefs, there's a very strong relationship between DIC and TA. And these graphs here, these are called the phase diagrams. If you're not familiar with them, you have DIC on one axis and alkalinity on one axis. I think about it in this way. Other people kind of flip these two. But in any case, the background colors are pH. And so for any given temperature and salinity, if you know the alkalinity and DIC, you can know the rest of the carbon system. And so you could calculate pH. You can also put saturation state, PCO2, and anything else behind there. And then by knowing how a biogeochemical process changes this ratio, we can see how it affects things like pH and saturation state in the background. And so, for instance, calcium carbonate dissolution and calcification change it in a 2-to-1 ratio. We're assuming this is our starting point. Respiration and photosynthesis generally about a 0-to-1 ratio, but this depends on nutrient uptake as well. And so these ratios throughout this global meta-analysis range quite significantly from really low. And this TADIC slope, or this ratio, is indicative at lower values of more photosynthesis and respiration. Or at higher values, we have potentially more calcification and dissolution. And so this balance of these two processes was really interesting in trying to what we were pull apart in that paper. So that's sort of the introduction to what I'm going to talk about moving forward. But that paper made me start to think about a lot of things as far as coral reefs and carbon metabolism go in my work. And so there's three guiding questions. I'm going to hopefully get to all three of them in 30 minutes here that really I thought about when putting together this presentation. And the first one, is there a reef-wide metric? So can we actually use something like this TA to DIC slope that can inform things about coral metabolism and reef function? And then as I did some projects into that, you'll see how all this comes together. Wanted to know how different components of net community calcification. So net community calcification or net ecosystem calcification is telling us about a group of organisms, but can we tease apart those different organisms? So that's sort of the second question I'll touch on. And then the third one is, it turns out coral reefs are not isolated systems. They're generally estuarine systems in the sense that they're coastal, right? And so there's lots of different processes that impact the chemistry of coral reefs, not just that simplified organic versus inorganic metabolism. So really thinking about the myriad of coastal biogeochemical processes and links between other systems, coastal systems, and coral reefs. So most of what all of what I'm going to talk about today really is based in two different areas of the world. So one place was Lizard Island on the Great Barrier Reef, and you can see here it's sort of at the northern end of the Great Barrier Reef. And the second is the Florida Reef Tract. So Great Barrier Reef is the longest barrier reef in the planet. Florida Reef Tract is the third longest, but they are quite different in their makeup and general state of health to begin with. So the first sort of story I'm going to tell you, we're gonna start on Lizard Island, and in particular, we're going to look at this reef flat here. And this reef flat is circled here on the map of Lizard Island. You can see the sort of lagoon that is around the island. This reef flat has received a lot of attention from biogeochemists because of the way the water flows across it, which I'll explain in a minute, which allows us to measure coral metabolism. So it allows us to measure carbon and oxygen fluxes as the water flows across this reef flat, generally from offshore here to inshore in this direction. One of the main reasons we ended up going to Lizard Island, I received a fellowship, a postdoctoral fellowship at the time from the Lizard Island Foundation to go do some research there. And we were interested in how this reef flat recovered after a multitude of different disturbances that had impacted it. And these impacts were two tropical cyclones in 2014 and 2015, and then a mass bleaching event in 2016, 2017 that really decimated the coral cover. This is showing you the impacts of cyclones and the impacts of bleaching on the coral cover and algae cover increasing during this time period. So our question was really based around whether we could measure a biogeochemical signal that reflected a recovery from these disturbances. The reason we chose that was, well, there were disturbances there, but also there is a lot of historical data there. This reef flat has been very well studied, and in fact, pretty much what I would call the Bible of my PhD thesis, which was the PhD thesis of Barnes, was actually Kinsey, sorry, Kinsey, was really kind of what started the work there. And they did this back in the 60s and 70s, looking at how chemistry changed as water flowed over this reef. And so what we did in 2018 and 2019 with this was with a group of collaborators out of different institutions, who I'll recognize at the end here. But we were interested in measuring the changing chemistry as it flowed over the reef here. And basically what you're looking at here are general flow patterns. So the directions are the way these arrows are pointing, and the colors are different speeds. And you can see flow generally goes from offshore to inshore here. And so what that allows us to do is measure the chemistry offshore and the chemistry at these two stations. And then we can understand basically residence time, how long this water took to go from here to there. And we can get flux rates from this. So this is flow respirometry. It's probably one of the older ways to measure metabolism in coral reefs. In general, the flow is going in this direction, although there are times when tidal forces take over and sort of move the water around in different ways. But when we did this, we had stations out there that were auto sampling carbon chemistry data every 2 hours, and then we would bring these back to the lab and measure alkalinity and pH in the lab. So this is what the sort of raw data set looks like. It's a little bit difficult to see, but in black here is the offshore data. So we took offshore data as much as we could, but that was really dependent on local conditions. But it's generally flat, although with some sort of minor, sort of longer term changes. And then you can see the change in the chemistry onshore, where negative are decreases in alkalinity indicate calcification, and then increases in alkalinity indicate dissolution. So if we put all this together with the flow data, we can develop net ecosystem production and calcification metrics, which you're seeing here per hour. And these are from the 2 years that we went there at the lower site. And that was really, you can see increases in production and calcification with light in the background during this, but this is the raw data. And really, we were interested in, well, how did this change over time compared to the previous studies? And so the first thing was to just look at the general changes in daily calcification and production and compare it to the 75 data and then some of the other studies that had been done. And one of the main takeaways from this for what I saw was that there's a huge range in calcification and production. And it even matches some of the earlier data from the 1970s. And so this signal that we sort of were expecting to find, which was a really decreased calcification rate because of all these disturbances, was not really there. And in fact, calcification was at similar rates potentially for what was going on back, a few decades earlier. And this really brought about the question about how do we compare data from these metabolic measurements across time when there's so much variability from day to day and hour to hour. And so one common way to do this in the Reef literature, at least, is to create these daily composite plots, which you can see here, where you just take the average rate at a given time of day, and then you can plot it against time of day. And here we have net ecosystem production, net ecosystem calcification, and you can see a strong daily signal where you've got net respiration at nighttime, right? And photosynthesis during the day. And then maybe some hints of dissolution at night on the reef, but really increases in calcification, which are well known under light conditions in corals during the day. And these are from the two expeditions we did, and you can see how well that the calcification sort of matched each other between these two expeditions. And then if you look at the production, you might see more respiration during the 2018 expedition. And so if you plot that against some of the previous data that had been done looking at how these rates had changed through time, you can see that despite maybe seeing some changes in the mean, if you just look at this across all these different time periods, you see lots of variability. So Silverman's data set was from the mid 2000s, and you can see a lot of variability. And really, these measurements were made pretty sparingly, maybe 15 to 20 measurements, because it's really difficult to make these measurements, to be out there at low tide. Logistically is quite difficult, and then to sit out there and take 24-hour measurements is quite hard. So really, these daily curves are based on not a lot of actual samples. So one of the things we wanted to do, and this was led by a postdoc at the time, Irene Hugh with Yui Takashita at Mbari, and Rebecca Albright, who is at Cal Academy. So these were the two main collaborators for that trip. She was interested in looking at a statistical analysis. Well, if we take a number of data points that we measure for this NEC and NEP, how many do we need to get till we get to that same daily sort of curve? And so she did some statistical analysis and showed that in order to get within sort of this nice range here in between these two brackets, you almost need to get up to sort of 60 to 80 data points. And that's way, way more data points than really have been measured in previous studies. So it makes it really tenuous to compare previous metabolic rates to modern metabolic rates. And then really why we want to do that is to assign sort of changes in things like ocean acidification and other global processes and local processes to metabolic changes there. So that brought about a second way that I thought is really probably the way that we need to move forward to compare rates over time, and that is looking at these photosynthesis and calcification irradiance curves. So this is the net ecosystem calcification on the right and production data from these curves. And you can see the raw rates in little dots in the background, and then you can see the binned averages for the different par levels in the bolder, bigger circles. And you can see that NEP and NEC both follow this distinct photosynthetic irradiance curve where they increase as light increases until they reach some sort of saturation value. And these are really useful because you can pull different metrics out of them, which I'll talk about in a second. But these metrics can actually tell you about different things, how these different metabolic activities are related to light. And so you're sort of normalizing what's going, the metabolism to the light levels. And so say you went and measured at 1 year and it was sunny the whole campaign you're out there, and you went and measured the next year and it was cloudy. This helps to sort of get around the problem of comparing those rates. You can't really just compare the average. And so this sort of stimulated this project, which was run by Jenny Milan. She was a PhD student at University of Glasgow at this time. She is now a research scientist at Nova Southeastern University, and I'll talk about another study we're collaborating on together. But for this study, Jenny was down at Moat in Florida, and she took coral and CCA. So here we have different species of corals and two types of CCA that she did incubations on. So these little pucks of these guys, looking at metabolism under different light levels. And so she came up with these PI curves. So this is production with DIC versus light, and you can see the same sort of general pattern where you have increasing metabolism, negative respiration at night, increasing metabolism that stabilizes once it reaches the saturating light level. And so this is really well known in a lot of plant and other, and even in coral reef sciences. But what is less well studied in corals is this relationship between GNet and PAR. There are quite a few studies out there, but comparing all of these things together, there's not many studies that are looking at how they interact with each other. So how light, photosynthesis, and calcification are all interacting on this organismal level. And so here you can see the curves that she got from the different corals and then the two CCA. And in general, if we just zoom out and we look at the rates plotted against each other, so here's photosynthesis or production, here's calcification. You can see there's a relationship between the two that is different between the daytime, which is over here, and the nighttime. And what we sort of hypothesized was going on is that this is related to energetic balances. So corals that are respiring more at night also are generating more energy for more calcification, whereas corals that are photosynthesizing more in the day are generating more energy, more calcification. So I really think that this is an energetic sort of balance within the coral where these two processes are related to each other. And so you can get a bunch of coefficients from these different PI curves. So Pmax is the saturating light levels, or it's the saturating photosynthetic level. You get this alpha, which is the slope, so how quickly you're rising to Pmax, and you also have your respiration, or in this case of calcification, it would be nighttime calcification or dissolution. So if we look at the rate of basically so Pmax or Gmax versus respiration, so this rate versus respiration rate, we see that same ratio where we see a increase in calcification as R increases for each of the organisms. And then opposite, we see the same across the different species. We see Pmax and Gmax increasing. So sort of just verifying this graph from the variables that we can pull out of these photosynthetic irradiance curves. Now, another thing this study brought up that was quite interesting was can we look at this ratio, this TA to DIC slope, from these individual corals and pull out any information? And I was interested, do we get a static sort of percent any NCP or percent amount of organic metabolism to total metabolism from these individual from these individual organismal incubations? And so to do that, this is the Lizard Island data, and this is sort of what piqued some of my interest on that, is that if we took NEC to NEP during any given time of day, we got some really weird things that were happening. So this is the ratio of calcification to production. So this is really what should be related to that TA to DIC slope that I started the whole talk out with. You get some really weird things that happen. Number one, you get some crazy numbers. But when you zoom in, you can see that at nighttime or at dusk and dawn, things start to go a little wild. And this is because this ratio starts to approach zero as NEP shifts from positive to negative overnight. So Jenny and I thought that a new metric might be a little bit better to look at this, and we called this the calcification to total carbon metabolism balance. And so what it is the absolute value of calcification to the ratio of calcification plus respiration. And this value goes between 0 and 1, so you sort of get rid of all of this craziness at this dawn and dusk times. But you can see as it gets to, so this is light curve, so this is par versus this GNet to M total metric across these different organisms Jenny incubated. And you can see at low light levels, you get this peak. And that is just related to the point where PNet is going towards negative, but GNet is still staying high. So you kind of reach this time point where GNet is high and dominating the whole carbon cycle. Now, this brought some interesting questions to me because from our study that I published in Plus One, claiming that we could find one single metric for these reefs to tell us about the ratio of calcification to production, I started to question that because this ratio was not static throughout a light curve. Why would we expect it to be static in a system where a bunch of different organisms are calcifying and you have all sorts of other things going on? So going back to the data from Lizard Island, this slope of the data we collected from these two stations, right? So this was indicative of what we were calling the metabolic balance in this Plus One study. And it finally dawned on me that this value, this slope, is this sort of change between the values that are on the reef flat itself. This is our average sort of starting conditions, TA and DIC. And the real slopes that we're interested in are these slopes from the starting value to the end value, right? This slope is actually telling us that ratio of NEC to NEP at any given time, while this ratio is telling us something a little bit different. And you can sort of see that here with the slope. Whoops, sorry. The slope here, and then this is the probability, the histogram of all the different slopes you get, and the star is the slope here of the entire thing. And it's not really matching up to the average slope or anything like that. So it's not necessarily telling us much about anything about what's going on. So what's going on there? I made a model, and I'm going to run through this super quick, so bear with me. But it's just a simple box model to look at how you can alter NEC and NEP and how that affects this TA to DIC slope. And really, the box model did a couple things. It kept NEC in blue, so calcification constant, while we altered NEP over the course of a day. And it did the opposite. It kept NEP constant and it altered NEC over the course of the day. And then for each time point, I plotted an NEC to NEP, or I mean a TA and DIC ratio from the box model. And you can see based on the colors, you've got when you kept NEC constant but altered NEP, your ratio was saying that everything was photosynthesis, right? Vice versa, when you kept NEP constant and moved NEC, the opposite, the ratio, the slope of this data went towards 2. And so what that indicated to me was that this ratio is not really telling us about the ultimate ratio of NEC to NEP or this metabolic balance of the reef. It's telling us about how these two things are changing over time. And so this little video sort of explains this a little bit better. You can see as you're going through these different time periods here, you can see that as NEP and NEC change with each other, that's how you get these different slopes. And so really what these slopes between TA and DIC are telling us is more related to how NEC and NEP change over the course of a time period than they are the actual metabolic balance. Which brings into question how we can actually measure these things in reefs. And so is there a reef-wide metric? I'm not convinced that there is one that's at least easily measurable. I think we need to measure these metabolic flux rates and we need to look at them against a light curve, honestly. And that's not easy to do. It's quite difficult and time consuming, but ultimately I think if we want to get comparative data that we can look at through time and space, those are the metrics that we need to be moving towards measuring as a community looking at these things. All right. So the second question, and these two are a lot shorter, but I'm going to go through them pretty quick because I'm coming up on time here. The second question was, can we tease apart different components of net community calcification using geochemical techniques? And part of what spurred this, so while we were out on Lizard Island in 2018, Manuela was running some experiments. So she was a postdoc at Carnegie at the time with Ken Caldera. And she did an experiment where she took these kiss-me's. So these are basically little incubation chambers that measure the change in oxygen and change in alkalinity. And you do in situ incubations on a live coral, and she did a dead coral as well, or a recently dead coral. And what we found was that production and calcification, so dead is in circles, and calcification was quite high even on the dead coral. So what's going on? Obviously, the dead coral is not calcifying. It's probably some sort of inorganic precipitation that's being driven by photosynthesis of the benthic algae there. You can see the coral maintained calcification at night while dissolution was occurring in the dead coral, the dead substrate. So this sort of brought about the question of, what is contributing to these net ecosystem calcifications at rates? And if dead coral is contributing quite a large amount, what are they actually telling us about reef function? So can we actually tell different functional groups apart? And so this was another project that Jenny was working on, and now she's taken over. She's now a Smith Fellow at Nova Southeastern and a research scientist there. So this is after her graduating from her PhD. And what she did was a similar thing to the previous study where we took different types of organisms and really here just CCA and coral and incubated them to try and see if we could use this sort of semi-novel technique that was really inspired by Zvi Steiner, who used this on the Red Sea. But basically, aragonite and high mag calcite take out a different amount of strontium to calcium. We thought we could do this with magnesium, but the background magnesium concentration turns out to be too hard to figure this out with. But with strontium to calcium, high mag calcite is taking out less strontium to calcite than aragonite. So we wanted to see, could we measure those changes in the water column? Are they reflected in changes in water chemistry in strontium and calcium? And so Jenny incubated 100% CCA all the way up to 100% coral. And this is really just some preliminary results. But it looks like, so if we look at the change in strontium over the change in calcium in those incubations, they're sort of matching up with what we would expect from 100% CCA all the way up to the 100% coral, following this nice gradient. So indicating that this could potentially be useful as a technique to look at the differences, communities that are calcifying. And this is important because CCA and corals provide different reef functions. CCA are sort of the glue of coral reefs. They also are settlement areas for baby corals, where corals are providing that habitat structure. So paired with alkalinity, I think this is a promising technique that we're still sort of working on. We did some Kiss Me incubations that we're going to hopefully turn into a paper in the near future with that. Okay, so then we're going to come up to the third question, and this is just going to highlight some of the work that's going on in my lab here at Georgia Southern in collaboration with some work down in Florida. But what role do the myriad of coastal biogeochemical processes play? Lizard Island is a very special place for the physical dynamics and the fact that there's a nice on to offshore gradient. But can we actually use this in other ecosystems? How difficult is that to use? Whoops. So I lost Carissa's picture, but she's on the next one. So this is a study that was led or is led by my PhD student, Carissa McPherson. And she did some measurements of the water chemistry here in Florida Bay. And she was interested in how seagrass impact the chemistry, but basically found the variation in alkalinity seasonally in Florida Bay to be quite significant, with higher alkalinity values in the winter. And here you can see alkalinity versus DI or alkalinity versus salinity. I don't know where my axes went here. They were there earlier. And you can see that during the summertime, you've got much higher, lower alkalinity values, but higher salinity values, and vice versa during the winter. But you have this negative relationship indicating that some sort of freshwater inputs, be it groundwater or surface water from the Everglades, are highly influential in the chemistry here. So Carissa just recently got for an Everglades Foundation grant to explore this a little further. So we did our first sampling expedition a couple of weeks ago, and she's collaborating with Heidi Hirsch, who's a research scientist at NOAA AOML down in Miami. And we're interested in the connectivity between Florida Bay and the Reef tract. So looking at how the chemistry between these two systems is interacting. And this is really based on a model that Heidi's been working on that was developed with two of her collaborators, Thomas Dobelaire and Emmanuel Hannert. And they developed this SLIM model. So it's a 2D hydrodynamic model of the whole area here that can track water movement. And we're interested in how chemistry was affected across these transects that they go out to every month. So this is her paper looking at that. Basically showing that in the southern Keys, if you tracked water back from these different transects, you got water chemistry, or you got water signatures that were coming from the bay itself. This is showing basically that same thing with now Carissa's data points put on there. And you can see that there's a clear indication in the southern Keys, or the mid to southern Keys, that water from the bay is coming out through these channels and it's impacting these reefs. And this is another model run Heidi did, showing how after 10 days, releasing particles at the sites where Carissa had done her original experiment, that water is eventually, after 10 days, some of that water is making it out onto the reef flat. And so this brings about the question about what do we use as the end member for the chemistry changing on Florida's reef flats, but also thinking about these systems as we get into these bigger sort of biogeochemical systems that become a lot more complex, and we can't just simplify this carbon fluxes into net ecosystem production and calcification. And we've got to think about everything else that's going on inside these really complex estuaries. So that sort of ends that question. And I think right now I'll leave this up for discussion because I think I went a little bit over the timeline there, but I just want to thank everybody that was involved in this, and hopefully that was informative and can presentation and can generate some good discussion. Thank you.